have our carrier proteins. And these are going to change shape. So I think of it as just when we're carrying someone, if I was going to pick you up, let's say this room is our cell. Okay, we have that guy sitting out there. Let's say we want to bring him in, and we're going to use a carrier protein to do so. If I'm the carrier, I'm going to have to go, and I don't just like with my mind, pick him up and move him, like the leak channel might. He just kind of moves on through, like the door's open. Carriers, we're going to have to go. I'm going to have to pick him up, bring him over here, and drop him, right? So this one is going to change shape. So let's say this is my molecule that we want to bring into the cell. And this is also the cell membrane, right? This kind of goes all the way around. The plasma. Goes all the way around.
What is that? Sorry.
This is going to be, the phospholipid bilayer makes up our cell membrane.
Oh, okay.
So these are going to be going all the way around to give you a 3D effect. My arch scales. So this one is going to open up. It's going to bring this in here. Once it's in here, we're going to close this gate because only one side is going to be open at a time. Then we're going to open this one and let it come out. So kind of in this picture, we see the same thing on page 63. Is we have here, we have our molecule being transported. We have our carrier protein that's going to go in there, here, and notice this closes here. And the carrier protein, which is right here, is going to change shape and releases the transported molecule on the other side of the plasma membrane. The carrier protein then changes back to its original shape. Then it goes back here and brings in another one. So that's carrier. Does that make sense?
Mm-hmm.
Okay.
Now, are the different molecules that come in, are they all different shapes, or you know what I mean? Because you said it changes shapes.
Changes shape as in this one changes shape to
Okay.
fit it.
So those are.
This doesn't really change shape. Mm-hmm. So this one is the same, but this, let's say this has to go to this shape in order for that to fit in.
Okay.
Okay, so then there's a few things that are going to have to do with our carrier proteins. And do you guys remember what they were? It was competition, saturation, and it's like selectivity. Selective, so basically it's selective. So we have selective, saturation, and competition. I think we have a good picture of this somewhere in here. Maybe not. But, so what do you guys think? Oh, here we go. So this figure on page 71 is going to show you. So we have our specificity. Is that how you say that word?
Mm-hmm.
I'm not an English major. Specificity is this one. So it's going to be specific. It only wants certain things into the cell. So notice this one says yes, that one says no. So only molecules of the right shape are going to be let in. So let's say this one, we have this molecule, and we have this molecule. These are all going to have different carrier proteins for their purposes. So let's say here's another channel, right here. This one is going to like star shapes, or whatever that blob I draw. So this one kind of be, let's say for forms like this. Kind of see what I'm getting there?
Yeah.
It's kind of going to want that to come in and out, but only one side's going to be. So this one is going to be like, yeah, sure, come in. And if this one tries to come in, it's like, sorry, I'm not the right shape for you. I can only pick up someone so big out there. I'm not that strong. So maybe I'm going to go for the littler people to bring in. Whereas someone else, I have this big guy who's the carrier protein, he's going to leave all the little people, or the people that are the wrong shape for him, because I can get those. He's going to go get the big monster men, who are at the gym all the time.
Okay.
So that's going to be specificity, okay? Does that make sense?
Mm-hmm.
Where's this one? See, this one's a triangle one. That'll go one is going to be saturation. And that is, I was talking about competition. Saturation is just the fact, see where these pictures kind of come in handy?
Mm-hmm.
If you know the pictures, you'll know all the information you need to know. Yeah, saturation. So right here we have one carrier protein, or actually we have two. Only if there's only one molecule that needs to come in, obviously we're going to bring it in. If there's only one guy out there that I'm trying to bring in, I'll pick him up, dump him here. Even, let's say we're working as a team, so we're all carrier proteins. If there's only one person out there, one of us will go, change shape, dump him in the room. But then let's say there's two guys. So now two of us are going to leave, pick him up, drop him in. What happens when, let's say there's a whole class, there's a lecture hall full of people? We can only move so fast, right? So we're all going to be saturated. These are our proteins. Remember, only one side opens at a time. So let it in and then release it. But we can only move so fast, which is why on this chart, they're going to draw it like this. And number one, when the concentration of molecules outside the cell is low, the transportate is low because it is limited by the number of molecules available to be transported. Right? So I only can bring one in at a time because there's only one person. It's not like we're going to be moving super quickly. But when we have, let's say, 3, it's like the perfect number, we're going to be moving very quickly. There's enough things to bring in, this room's going to start filling up. But, so then it's going to spike. Right then when we have kind of an equal number on both sides. And then it's going to kind of plateau when we have a whole lecture hall because we can only move, we're just moving the exact same, at the same rate as when there were only 3 people that we had to bring in. So that's going to be our saturation. And then competition is exactly what it sounds like. There was a picture on this page. It's just whatever gets there first gets to come in. So, let's say they're both the right shape for this one. This is going to be our little star guy again, or blob. The law of specificity says they both can come in. We're both accepting of them. But whatever one gets there first, this one gets here at 8 a.m. This one comes at 2 p.m. Which one's going to get to come into the cell? This one's going to go through the carrier protein.
The other one can't come in later?
It can, but it's going to be the law of saturation. So maybe now it takes longer to come in.
Oh, okay.
Mm-hmm. So that's competition. Saturation was, sorry, we're full. I'm only one person, I can only pick up one person. Maybe then they'll go find another one and then be able to come in.
Okay.
And then selectivity or specificity is just, this is the right shape. Ta-da! So that's
they
our
kind of overlap each other.
Yeah, they all kind of work together. So you can say that one thing could have all 3 laws in the exact same situation.
Okay.
So like this one would be one of them. It's the right one, so we're selective. We got it. Saturation, sorry, we can only take so many. Whoever's get theirs first, competition is going to come in.
Okay.
So that's going to be, so we have, which ones have we gone over? We have the channel, which is a good example of the leak channel. It's going to go from a high concentration to a low concentration, which is diffusion. When I spray the perfume, it'll go out through the cell.